Thank you for coming to support the Marine Life Talks here at Knox. As a second year PhD student at the National Oceanography Centre, a little bit about the research I've done so far during my PhD with a talk entitled Dinner in the Deep Sea, Organic Falls from Contrasting Environments. So just to tell you what we're going to do today, first a little introduction on organic falls, and then tell you about two of the main groups of fauna found at organic falls, Oceanography and Seafloor. Then we're going to go into my first chapter where I investigated a natural whale fall in the Southern Ocean, and then tell you about one of the main types of fauna found on this whale fall, the Antarctic ocedax. After that, I'll touch on a brief trip to the Bahamas and some other work from off South Africa, and then finish with why we should study organic falls in the first place. So, many of you come to these talks quite often, and so you've heard quite a bit about the deep sea already. But just to reiterate, this is what the deep sea typically looks like. As you can see, there isn't an obvious amount of life, huge amount of life. But this is because it's really dark, it's really cold, there are very high pressures, and there isn't a huge amount of food in the deep sea. Most of the food which arrives on the deep sea floor comes from the sea surface, thousands of meters away, in the form of dead plankton. But occasionally, we do tend to get much larger packages of food drifting down to the deep sea floor, and these are what we term organic falls. So, they can be anything from a swarm of jellyfish to a piece of kelp, even to be a bit morbid, a human body. But the two most spectacular types of organic falls are the two types of falls which I happen to be studying, which are whale falls and wood falls. Pretty self-explanatory. And when whale carcasses and wood arrive on the deep sea floor, there's a huge source of food to animals in what is usually a food-limited environment. So one whale can be about 160 tons of carbon. So that's quite a lot. So when they arrive on the deep sea floor, they tend to prompt this kind of feeding bonanza. It's like it's Christmas in the deep sea. Animals come from far and wide to feed on this carcass or piece of wood. And not only do they provide food to animals, but they also provide shelter and some hard substrate for many animals to settle on. And because they have so many uses to deep sea fauna, they tend to create these ephemeral island-style habitats, this island of life in what is usually quite a deserted deep sea. Depending on the size of the organic fall, they can last from anywhere from a few days to a few years, and it's thought that some whale falls can even last up to 100 years. But the most amazing thing about organic falls, which a lot of people don't realise, is that they are actually chemosynthetic ecosystems. And so a few of you may have heard of hydrothermal vents or cold seeps, and these are possibly the most well-known chemosynthetic ecosystems. What chemosynthetic means is that instead of using solar energy as the main form of energy, we now use chemical energy. And there aren't that many ecosystems in the world that can do that. But whale falls and wood falls are two of them. So, when a whale carcass arrives on the deep sea floor, it tends to go through 3 stages of decomposition. The first stage is known as the mobile scavenger stage. And this is when the carcass is fairly intact and many large scavengers move in to eat the flesh of the whale. So you tend to get things like 6-gill sharks and other deep sea sharks, hagfish, rat-tail fish. I'm not really sure what these are meant to be. Maybe some kind of crab. You do get crabs, lobsters, and amphipods and isopods. And this stage can last from anywhere, depending on the size of the carcass, from a few days to maybe a year or so. And this is a little video of one. So this is a grey whale, which was actually implanted by scientists off California. And this, as you can see, is clearly the first stage, as there is a huge amount of flesh left. This is a 6-gill shark, just trying to get in there and make the most of this amazing food resource, which has suddenly appeared. And you can also see lots of hagfish floating around as well. I think he looks like he's smiling. He's really happy. So The next stage is known as the enrichment opportunist stage, and this is when most of the flesh of the animal has been removed, and the sediments surrounding the carcass tend to become enriched with organic matter. And this organic can come from feces from animals that may have been feeding in the previous stage, also animals that may have died or been predated upon when they were feeding on the flesh, or even leftover bits of whale tissue, small pieces that have gone into the sediment. And during this stage, you tend to get a completely different set of fauna moving in to exploit this food resource. And this set of fauna tends to be much smaller in size than compared with the mobile scavenger stage. So, little video, to go back. You tend to get a lot of bacterial mats, thick white bacterial mats, which form all over the sediment. You also get things like isopods, amphipods, and even lots of types of polychaetes. And you do still have the occasional scavenger hanging around, like a rat tail or a hagfish. So this is the same whale that you saw previously, just 18 months later. You can see there's very little flesh left. The occasional hagfish hanging around, but you can't really see any real fauna, and that's because they're quite small and possibly within the sediment. But in a moment, you'll see some aggregations of polychaetes. So these are actually a species called Vitoniella flocati. And for any of you that know anything about carpets, which you know is an important thing to know stuff about, flocati is actually a type of rug, which is, I think, wool and handmade, a shaggy rug. And so they were named Vitoniella flocati because they tend to do this weird thing where they hang down from the underneath of a bone and sort of form like a rug-like thing. So, yeah. And then the third stage of decomposition is known as the sulfophilic stage. Sulfophilic means sulfur-loving. And during this stage, there is no tissue left on the skeleton. And what tends to happen is that whale bones have huge amounts of oils in them, huge amounts of fats in them. And that's part of the reason they were so prized by whalers about 100 years ago, for these oils within the bone. And these oils are used by bacteria which respire anaerobically, so without oxygen, for food. And as a result of this reaction, a byproduct of this reaction, sulfides are given off. And these sulfides are what form the chemical energy for this chemosynthetic ecosystem. So instead of having solar energy as we do on land, we have chemical energy in the form of sulfides. And instead of having plants as we do on land to use the solar energy, we have bacteria which use the chemical energy and are the primary producers during this stage. So it's like these bacteria mats here. You get these thick white bacterial mats which use the sulfides as their form of energy. And then they are what most of the animals that come during this stage will feed on. And then also lots of animals tend to have bacteria within them which can use the sulfides and then just give them a direct source of food. So during this stage you tend to get things like mussels, which can be chemosynthetic, living in the sediments, other gastropods like limpets and so forth, living on the bones themselves, amphipods and isopods still hanging around, lots of polychaete worms. And because, as I said, food is really scarce in the deep sea, animals have evolved to make the most of what they've been given. So we even have animals during this stage which will consume the bones of the whale. And that's what oceanids do. So oceanids are a type of cibberglinid worm. So they're most closely related to hydrothermal vent tube worms, so the big ones with the big red plumes. And as I said, they come in the third stage of whale falls and live on the bones, as you can see here. So they've got this is an adult specimen, and you've got this trunk here, which tends to diverge into these four very beautiful palps, which are used to extract oxygen from the water column. Then you've also got the oviduct, which shoots their eggs out into the water column. And then you've got this huge mass, root mass, which they use to burrow down into the bone and suck out the nutrients which they eat, in a similar way to which a tree's roots would burrow down into soil and take up the nutrients. And so you can see here what they look like in situ. You can't see any of their roots. So on the roots, they have these unique bacteria, two types of bacteria. One which secretes acids to help them break down the bone, and then another type of bacteria which actually consume the collagen in the bone, which is what they eat. So they were only found, this entire genus was only found in 2004, so really, really recently. And within that time, they've been found inhabiting waters from 30 meters to 3,000 meters, but they tend to be mostly deep sea. And it's thought that they occur worldwide, but again, we haven't really found that many of them. But we think that they occur wherever whale bones occur. So I guess they'd be more prevalent under, for instance, migratory routes of whales, where there may be more whale carcasses coming to the seabed. But I've sort of forgotten about woodfalls. So to move back to woodfalls, at woodfalls there is also a type of animal which is incredibly abundant, and that's called xylophaga. And this is actually not a worm, this is a bivalve, so similar to a clam. And you can see one here, it's got these two shells, one, two, which open up and this massive siphon sticks out of. And it tends to burrow down into the wood, as you can see here. And usually you just see the siphon. So all these little things that look like plants aren't actually plants. They are the siphons of xylophaga sticking out of the wood. And these xylophaga burrow down into the wood, and in a similar way to the ocedax, they use bacteria to heterotrophically feed on the cellulose in the wood. And we think that they occur worldwide. We know that they occur worldwide. Apart from the Antarctic, there's never been a record of them in the Antarctic. And we know that they occur from 0 to 7,000 meters depth, but again, they're mainly a deep sea species. So we're talking about a worm and we're talking about a bivalve, two completely different classes of organisms, yet they are still quite similar. They're both mainly deep sea fauna, and they both occur at organic falls. One of the main things is they both bore into their substrate for nutrition. Ocedax with bone and xylophaga with wood. And they do this via bacterial symbioses, which they also feed with at the same time. And they also, because they bore into the substrate and eat the substrate, they have a huge effect on the wood and the bone which they're living in. And so we now know that ocedax are probably the main animals to break down bone in the deep sea. We don't think there are any others, really. And so they'll have a huge role in carbon cycling, for instance. But one of the most amazing things about these animals is that they exhibit something called sexual dimorphism. And that's when there's a huge discrepancy in the size between the male and the female of a species. So in this case, for both of our species, genera rather, there are large females and dwarf males. So with the ocedax, all the pictures I've shown you thus far have all been females. And these little blobs, 1, 2, 3, 4, those are the males, adult males. And basically they just stay little blobs all their life and attach themselves to the female, give her sperm until they run out of energy, and then they die, and then they're replaced. And similar to the xylophaga here, you can see the little male sitting on her back. Same thing. They just stay there until they run out of energy, giving her sperm, and then they die. And she gets new ones. If only it was like that in real life. And the last similarity is that once these animals settle in their adult form in their substrate, they don't move. They will stay in their substrate until they eat it completely, and then they will have nothing left to live in and die. And so the only way in which they can spread to new habitats is via their larvae, which they will usually expel into the water column, and then they'll be carried with the currents. So, moving on to my work. The first chapter of my PhD was investigating a natural whale fall in the Southern Ocean down here. And that was done using the James Cook. On her 42nd voyage, she went down to the East Scotia Ridge here as part of the CHESO project, which is the chemosynthetic ecosystems of the Southern Ocean project. And she was actually looking for hydrothermal vents down there. But during a dive with ISIS, our ROV, in this creepy seafloor caldera, she just stumbled upon a whale skeleton right here northeast of the subcone within the caldera. And it's at about 1,444 meters depth, so quite deep. And again, it was totally by surprise. So this is a little video of what it looks like. This is the vertebral column moving down slope. There are some ribs sticking off here, not many though. And then the skull is the furthest down slope. And you can see the bones, there's no flesh left, and the bones are very, very eroded. This is a jawbone. You can see it's fragmented against the rock. Just, it's obviously been there for some time. And then here are some fin bones. That's a humerus, and I think a scapula, maybe. So yeah. So upon stumbling upon this carcass, scientists on board thought quickly, and we decided that we wanted to sort of tell the story of this whale carcass. And to do this, we would need to do two things: sample some bones with the ROV, and also do some high-definition video surveys with the ROV. And so with the bone samples, I'd be able to DNA barcode the bones, and that would tell me what species of whale the skeleton belonged to. I also wanted to do something called a lead-210 radium-226 disequilibria analysis. And all you need to know from that is it would essentially tell me how long ago the animal had died, so how long the whale fall had been sitting on the seafloor. And I also wanted to identify what fauna was encrusting the bones to see, whether there could have been any new species, what was living there. And using the HD video surveys, I wanted to look at the state of the bones, how eroded they were, and that would give me an insight, give me and other scientists an insight into the taphonomy of a natural whale fall. Taphonomy means the fossilization, the process by which things are fossilized. And as you can tell, we don't usually find natural whale falls, and so this was quite a big deal and would give us, an insight into the decomposition of these animals. And lastly, I wanted to count the individual number of fauna on each bone, and then work out the individuals per meter squared of bone surface area. And with that, I would then be able to look at the abundance of each type of animal on each bone going through the whale skeleton, giving me a sort of estimate of distribution, microdistribution. And using that, I'd be able to test something called the oil gradient hypothesis. Still with me? Yeah. Unfortunately, the time since death didn't work, so we're not going to talk about that. But we're going to talk about everything else. So the oil gradient hypothesis, what is it? It was a hypothesis coined by a friend, Nick Higgs, and as I said before, whale bones have huge amounts of lipids in them. And what we now know is that it tends to change depending on the bone you're referring to in the skeleton. So the oil concentrations will vary as you move through the skeleton. So we're going to focus on the yellow, which is the oil. And you can see that it tends to be quite high in the jawbones and skull, and then decrease as you go into the cervical vertebra, and then it gets to its lowest levels in the thoracic vertebra, only about 10% composition. But then when you move into the lower vertebra, so the lumbar and the caudal vertebra, you tend to go back up to about 50% composition of the bone being oil, which is really high. So Nick hypothesized that because the lipids are what cause the sulfides to be released by bacteria, and the sulfides are what are the energy source for the chemosynthetic fauna during this stage, he proposed that the abundance of sulfophilic organisms during this whale fall stage will correspond to the changes in oil throughout the whale skeleton. So the most sulfidic would be the most lipid rich, and those would have the most sulfophilic fauna, so therefore the lumbar and caudal vertebra. But those bones that didn't have a lot of oil wouldn't have that much sulfophilic fauna on it. Yeah? And so because the bacteria tend to be the primary producers and directly use the sulfides, he thought that the bacterial mats could be used as a proxy for lipid content of bones. So that was what I wanted to test. This is a mosaic of the whale fall. You can see the skull down here. This is moving down slope. The skull, vertebral column. And you can see that a lot of the bones are quite displaced. Maybe because on a slope, I don't know how bones would roll uphill, but and there are quite a few bones missing. Only about 4 ribs left. A lot of the vertebra, which would have been hair, are gone. This is a fin up here. But because of the size, we think when the animal would have been alive, it was about 10.7 meters. And when I did some molecular work on the bones, So basically this is a phylogenetic tree, and all you need to take away from this is that each of these is a species of baleen whale. And so my sample is here. I took 2 samples from the bones. You can see they're the same level, which means that they're the same species. And they fit straight into this branch, which has the Antarctic minke whale. So that means that my whale is an Antarctic minke whale. And if we say the size is about 10.7 meters, we can say that it was definitely an adult Antarctic minke whale. And so, moving on to the bone encrusting fauna. From the entire carcass, we found 10 species of fauna living on the bones, all of which were new to science, which is very exciting. So all of these animals don't have names yet, and they're that new. So this is a new species of Pyropelta, a limpet, an osteopeltid limpet, and a lepetodrillicid limpet. And this limpet was actually found at nearby hydrothermal vents, so that shows that there's some degree of overlap between chemosynthetic ecosystems. There were also some pericarids, a lecyanacid amphipod, and this isopod called Jera. And what is exciting about Jera is that so far all records of any species from the genus Jera have been from the northern hemisphere, number 1, and have been from estuarine shallow environments. And here is this little guy from the Southern Ocean at 1400 meters depth. So he must have gotten really lost, I guess. But there were also 4 new species of polychaetes, 2 species of Ophiotrocha, and a capitellid, and also a brand new species of Ocedax. So you can see it here, these little red things sticking out the bone. Here's one of the limpets, and these white things are all the bacterial mats which grow on the bone. But yeah, they don't really do a lot, but you know, they're pretty. They destroy bones, which is cool. So here she is. And so this is the big root structures you saw before, the oviduct and her beautiful little palps. So when I looked at that and counted, went through all the footage and counted all the animals, I will explain this, don't worry. Basically, this lower graph is the bacterial mat abundance. This is the Pyropelta abundance, and this is the Ocedax abundance. Little pictures next to it so you don't forget. And each column represents a different bone. And underneath I've numbered the bones, and the numbers correspond with this diagram, and then the type of bone is above it, but I'll just talk you through it, because that'll be way easier. So, with the bacterial mats, we found that they were least abundant on any unidentified fragments, small fragments of bone, and least abundant also on the cervical vertebra, so the top vertebra in the neck, which just so happened to not have a lot of oil. But they were most abundant on the lumbar and caudal vertebra of the animal, so lower vertebra. The Pyropelta limpets showed pretty similar trends. They were not very abundant on the fragments and not very abundant on the cervical vertebra, in fact absent on some, but they were most abundant on the jaw bones and the sternum of the animal, so the chest bone. And the Ocedax showed a completely different trend. They were not very abundant on the lumbar and caudal vertebra, but they were very abundant on the jaw bones. And you can still see that there's quite a few in the cervical vertebra here as well. And there were also quite a lot in the thoracic vertebra, but I didn't include that in the graph because various reasons. So when I ran some statistics on it, we could see that there was a correlation between the bacterial mats abundance and the Pyropelta abundance, a positive correlation. So the more bacterial mats there were on certain bones, then the more Pyropelta there would be as well. And with the Ocedax, we saw a completely different trend. The more bacterial mats there were on bones, then the less Ocedax there would be. And so I have a few reasons why I think this is, which I'll tell you shortly. But when I investigated that relationship further with, for instance, this caudal vertebra, we can see here that these represent Ocedax and these are bacterial mats. And wherever the bacterial mats are found, there's never an Ocedax actually within where the bacterial mats are growing. So maybe that's because there's some kind of competition between the Ocedax and the bacterial mats, or maybe the bacterial mats secrete something which the Ocedax don't like. or maybe even, I have other reasons, I'll tell you shortly. So, when we look at the taphonomy of the skeleton, the first thing I want to say is that natural whale falls are quite rare. I mean, there's no way to find them really, unless you put one down there. And so to stumble across one, we do consider ourselves to be quite lucky, especially since industrial whaling began about 100 years ago. They are now much less frequent than they would have been 100 years ago. But when we did look at the skeleton, we could see that it was really severely eroded, which did lead us to believe that it was quite old. Especially the most eroded bones were the cervical and thoracic bones, so the upper vertebra. And these bones, the cervical and thoracic bones, tended to correlate with those bones where the ossidax were the most prevalent. So it's not really surprising given the effect which ossidax have on the bone matter which they live in. And then also there were lots of small bones missing, and this could be because maybe scavengers carried them off, or just currents took them away, or maybe they were totally sedimented or totally eroded. We'll never know. So, the main findings from this chapter. We can say with 100% certainty that it's an adult Antarctic minke whale the skeleton belonged to. And that there was a huge abundance of new species living on the skeleton. And that isn't really surprising given the fact that the Southern Ocean is a very poorly explored area. And there's a huge degree of endemism in that area because of the Antarctic circumpolar current, which tends to keep everything within that area. And then lastly, that we can say that we have found evidence to support the Higgs hypothesis that the lipid content does affect the distribution of fauna during the sulfophilic stage of whale falls. In that bacterial mats, which are sulfophilic, were found on the most sulfidic bones, which were the most lipid-rich bones, so the caudal and lumbar vertebra. We can also say that pyropeltalimpids, which happen to eat bacterial mats, mirror the distribution of bacterial mats. This makes sense. You want to be where your food is. And so they were also on the most lipid-rich bones. However, the ossidax showed a completely opposite distribution to the bacterial mats. They are found on the most lipid-poor bones. And these lipid-poor bones meant that they were the most eroded, so ossidax have a huge impact on taphonomy of skeletons. So, moving on to chapter 2. That little ossidax picture I showed you before, we got about 40 specimens of them, and we decided to do some DNA work on them. And basically, this tree shows all of the ossidax species so far, and then these numbers are all of my specimens. And when I ran the genetics, it turns out that my one ossidax species was actually 3. And because I'd gotten them in such bad condition, I couldn't actually tell from just looking at the worms that they were different species. So what was now one ossidax is now 3 species of ossidax. And these 2 are totally new to science, and this one has been found once before. And so this is where my whalefall was. And this is where the beautiful one was found previously. And between the 2 sites, there's 1,850 kilometers. So either their dispersal capabilities are amazing, or there are plenty whale falls along the way, which it can kind of stop off at. So, yeah, who knows? So now I'm going to digress from the PhD topic for a little bit and talk about a mini trip which I did. My supervisor at the Natural History Museum happened to get a small amount of grant money to do a project which involved a lot of public engagement. And so we decided, as we just purchased this little mini RV, which is only about this big, and can only go to 200 meters depth, that the obvious choice is to take him to the Bahamas. So we all flew down to the Bahamas to a very small island off of Eleuthera called Spanish Wells, and that's where one of Nick, the same student who cut his hand open on the whale, lives. So we went down there and decided to make these 3 packages. As I said, it was a very small grant, so it was kind of science on a shoestring. These were laundry baskets, which we'd bought in the market. and we decided to make wood and bone packages to test what would come and live on organic falls in a tropical shallow water environment. So we bought our laundry baskets, we got some coconuts as our wood, and some other kind of wood from the garden. And then we were donated a beached beaked whale head, which ended up looking like that. And we strapped them to our baskets, and the plan was, last October, to put them down at 20 meters depth, 40 meters depth, and 60 meters depth. And no one had ever done shallow water organic fall experiments before in this locality. So we just kind of wanted to see, on a whim, what we'd find. But we were really hoping that we would find the first tropical species of ocedax, because all ocedax that had been found before was from either very, very cold water or very, very deep water. So we were sort of hoping we'd hit 2 nails on the head at the same time and find a shallow water tropical ocedax, which we nicknamed Tropidax. This is representation of what I thought we'd find. So when we got our packages back, though, 6 months later, they didn't really look like what we put down. So it turns out that we sort of overlooked the fact that the Bahamas is actually a shark sanctuary and that sharks actually like to eat whale bones quite a lot. So we got back these perfectly cookie-cuttered laundry baskets and some teeth. Turns out this is the tooth of a tiger shark, and it has this unique shape, my supervisor told me, so it can get into turtle shells. So there's a little tip for the day. But anyway, this was just an example to show you that not always does science go according to plan, especially when scientists are silly and overlook facts like sharks like whale bones, and there are lots of sharks there. So yeah, anyway, back to the topic at hand, which is the PhD research, which does go according to plan most of the time. So just to touch briefly on what I'm working on right now, 2 years ago, there was a cruise an expedition down to the Southwest Indian Ridge, which is located here off of South Africa, which is here. And on that cruise, they deployed 2 moorings that look like this on 2 seamounts, 2 different locations. Both had some whale bones and some mango wood. And we decided to put these bones down because no one had ever really investigated any of the fauna in this area. It's a really poorly explored area. And also, no one had looked at organic falls from this area. So we just, again, wanted to see what we'd find. But again, we sort of had this little hope that we'd find an ocedax, kind of like this, grail. So last December, the James Cook went back, so 2 years after the packages were deployed, and collected them for us. And so these are really preliminary results. It's just to show you the abundance and the diversity of animals that you can get on organic falls. So there's everything from a basket star to some polychaetes, an emertian, gastropod, sea spiders. And these are just, one or few of each, but there are loads. Loads and loads and loads. And this is just a fraction of the type. And so when you consider that these are just packages about yay big, 4 of them, it just kind of shows you how important organic falls are to the deep sea in terms of being a resource for food, because so many animals come to use them. So, finally, why study organic falls? They're this silly little ecosystem, no one hears about them, you'll probably never see one with your naked eye, blah, 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 blah. Well, as we can see, they tend to harbour huge amounts of life. And a lot of this life, as we can see, does turn out actually to be new to us. And as cheesy as it sounds, we need to understand what lives in our seas in order for us to be able to protect it efficiently. And also, the organic falls tend to have species or families of animals which overlap with other chemosynthetic ecosystems, like hydrothermal vents and cold seeps. So it's actually thought that many of these fauna use whale falls and wood falls as sort of stepping stones. Because hydrothermal vents, for instance, may not be very close to each other. Animals may not be able to get from hydrothermal vent A to hydrothermal vent B, and so in one go as such. So the larvae will actually hop and then colonize the little whale fall and then hop again, and that's how they will actually get to their next main site. And they can do this because the ecosystems are quite similar in a lot of ways, namely that they are both chemosynthetic. And also, there's still so little known about organic falls. We barely know anything. And so, for instance, we now know that hydrothermal vents are actually, their main types of fauna vary biogeographically. So in the Atlantic, hydrothermal vents tend to have shrimp as their main fauna. In the Pacific, they tend to have tube worms as their main fauna. In the Antarctic, they tend to have crabs as their main fauna. And we just don't know whether these sort of patterns exist for organic falls. So, and we also don't know what kind of roles organic falls play. I mean, how important are they really to animals? We know they provide them with food, but if all the whales were to die in one go and then there were no more, what exactly would happen? Do they sequester carbon? You know, lots of these answers that we just don't know. And then by studying organic falls, we can actually learn quite a lot about the evolution of these animals and put this sort of like these animals into the context of the bigger picture, which is, you know, where do we come from? How did we get here? And did life actually evolve in the deep sea, as a lot of scientists do seem to think, at chemosynthetic ecosystems? And lastly, as with everything, humans always need to know that it will benefit them some way. So I'll leave you with that note. But basically, apart from the ridiculously cheesy, but still very important, finding new species inspires people and, you know, makes people happy, etc., etc. But a real benefit to society is that right now, Procter & Gamble, which is a really big pharmaceutical and everything else company, they've actually developed a washing powder where we all know that we need to try and cut our carbon emissions, right? Because of global warming, blah, 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 blah. So now they're telling us to wash our clothes at lower temperatures, right? And so Procter & Gamble actually got some of the bacteria which break down the lipids on whale falls and have used it to make a washing powder, a biological washing powder, which can actually wash clothes at the ambient temperature of the deep sea. So because these bacteria are accustomed to working at about 2 degrees centigrade, this washing powder can now wash your clothes at about 2 degrees centigrade. So I've heard they're not actually selling it yet, because we wouldn't be able to deal with the fact that we've jumped from 30 degrees to 2 degrees. And so they're actually staggering releasing the product so far. But I guess in a few years, you will have that washing powder and you'll be able to say when you buy it, I know where those bacteria came from. And that's it. Are there any questions?
Thank you. Fantastic.
No problemo.
Are there any questions?
When you went and incidentally found the whale fall,
Yeah.
You obviously hadn't been planning to do that as your research while you were there. You'd obviously been planning to do other research. So how much of a challenge was it to suddenly switch direction? Did you have to abandon your original research?
So I'll just repeat the question for the benefit of the recording.
Yeah.
So I think the question is, since you weren't planning to find a whale fall, how big of a deal was it for you to change directions on research when it's found?
It's a good question. I actually wasn't on the cruise. I was just given the PhD after. But my colleague Lee was on the cruise, and I think they were, as I said, they were surveying hydrothermal vents in the area. And so this was the first sort of expedition down there. And so it was really an exploratory venture. They didn't really know what they were going to find. And so the plan wasn't as concrete as many research cruises can be. And as far as I know, they were finishing the end of an ROV dive where they were doing a routine survey of this caldera. And they happened to see these white blocks in the distance and were like, well, what's that? And it turned out to be the whale fall. And I think you guys just dedicated, I think, 2 of the dives, and usually there are about 24 dives, maybe. And it wasn't 2 whole dives. It was just a few hours of 2 dives where they just did a routine, quite systematic survey, making sure they mapped the entire whale fall area. And then they picked up 3 whale bones on the other dive. And that was essentially all I needed to do with this much work. But I didn't say that we are going back in December on the James Cook to the Antarctic. And it is, again, the primary reason for the cruise is to visit the hydrothermal vents, which were found during that cruise. But we will get the chance to visit the whale fall again. And it is 2 years since we last visited. So there may be quite a lot of change in the animal, in the bone structure and what inhabits it. And also I'll get lots more specimens of the ossidax, so we can get a lot more pretty pictures, because the specimens I got really weren't very well preserved.
Were the bacteria the same? All the same?
On whale falls in general.
Yes, and the wood.
Sorry, the question, we can't have questions this way because the microphone doesn't pick it up. So the question is, were the bacteria the same?
Yes.
On whale falls and wood falls.
Yeah.
In terms of bacterial mats, they aren't the same species, but they do tend to be the same families or genus. And there'll be deep sea bacteria that just are there to break down organic matter. So I think the thick white ones are known as Beggiatoa, and they are quite prevalent on many chemosynthetic ecosystems, but it won't necessarily ever be the same species, but it will be the same genus or family. And between whale falls and wood falls, the same thing. It won't ever be the same species, but it will be the same family or genus.
In the front over here?
What do you usually do with a whale after it's been washed off?
Well, that's a really interesting question. So,
Do you want to just repeat it?
sorry, what do we do with a whale when it washes up? Well, right now, what we do right now is, there are 3 options, 2 options. Usually, if scientists can't get there in time, okay, no, let's start again. Usually, some scientists will get there and they'll try to find out how the animal died. That's the first thing they'll do. And they're usually from the zoo, the Zoological Society of London. Then after that, the whale is actually the property of the council on which the beach belongs to, of that county. And so to dispose of a whale is actually a pretty big task. So with that one that I had at the beginning of my slide with the big hole, they actually were trying to get rid of it after the film crew had done what they needed to do. And because they were so desperate to get rid of it quickly, they actually hired a tree surgeon to come with a chainsaw and try and cut up this whale. It didn't work because flesh isn't really that hard. And usually they just cut it up with big saws or something, and then they either use it to make landfills or just put it in a big dumpster and just kind of wheel it off somewhere. But what we as scientists would like to do is, which they do quite a lot off California and Japan, controversial, but when they get whale carcasses beached, scientists are given them to tow them out to sea. And because whale falls are not easy to find, they sink them. And it's sort of this in situ experiment, which they can go and revisit with an ROV and see the progression of decomposition. And that's how we know so much about whale falls, really, from towing them out, attaching loads and loads of weights, and then going back to visit them and see what happens. So either we sink it or we cut it up and put it into landfills.
In the back, sorry?
These creatures that are living in this great depth are accustomed to a very high pressure.
Yes.
What are the problems of studying them with regard to?
So in terms of, sorry, okay, repeat.
The question.
These creatures tend to be living at quite high pressures when they're at depth. So what are the problems with studying them? So in terms of what I tend to study, which are invertebrates, there aren't as many problems as with studying fish, for instance. Because fish have swim bladders, we know that if you bring them up from depth, the air in their swim bladder expands rapidly as the pressure decreases, and you end up with stomachs exploding out of mouths, or fishes just exploding, or whatever. But with invertebrates, when they do come to the surface, quite a lot of them are still alive. The ROV will pick them up, put them into the little box, carry on doing whatever it needs to do, and then return to the surface however many hours later. And when they arrive on deck, quite a lot of the time they are still alive. They are much slower, but still hanging on. And so I'd say the main problem really is we don't tend to keep them alive for very long. We'll put them in the lab, take pictures of them while they're alive if we can, because of course colours and stuff, once you preserve them, will disappear. But usually, yeah, they are still alive, and obviously with working at depth, we can't go there ourselves, and so one of the main problems with surveying at depth is that what you collect at that point in time, you may never get the chance to get again, and it's not particularly easy to go back, getting a ship all the way down to the Antarctic and then diving 2 kilometers down. Doesn't happen very often. But yeah, I guess it's not very humane to bring them up to the surface, as it's such a huge difference in pressure for the animal, and so as a result we just tend to take them out of their misery pretty quickly.
Two questions. The first one is about those two regions. If you found the wetfalls
Yep.
in 1850, is it correlated with the current regime in that area?
Okay. Oh, the two worm species.
Yeah. And the second one.
Okay, wait, hold on. Yeah, I'm listening.
In DNA analysis, you use the microsatellites to identify which they are the same species. Which technique you use on DNA?
Hold on. Okay. So first question. Wait, I passed it. What am I doing? So these two sites.
Yeah.
For those of you who don't know, there is a current which goes all the way around the Antarctic in a circle, and it's called the Antarctic Circumpolar Current. And it just continues around in this circle. And as a result, it acts as a sort of a barrier to animals being able to leave the Antarctic and enter the Antarctic quite a lot. And so I haven't actually looked into it because I'm now writing this paper, but it does make sense that we're not sure. I need to do some population genetics on them to see which one came first, but it is thought that because of the huge amounts of current that they could have just easily gone. I'm not sure whether the current goes that way or that way, but yeah, I would think that it was current that did transport them between the two, because there is this huge current which goes all the way around the Antarctic. And so maybe this species does actually, maybe all the Ocedax species do tend to be circumpolar as a result. But that research is still to come. And the second question was about the Ocedax microsatellites.
Yeah.
We use primers to isolate the same sequence of DNA and then just ran them in a tree, and that was it. 2 primers which were made for Ocedax. Okay? Any more questions? Hey.
Going back to your minke whale.
Yep.
You said that the bacterial mats use the oil as
Yep.
the primary source, and the Ocedax breaks down the bone.
Yeah.
So do you expect that once the oil is gone, when you go back, that the Ocedax has moved to the bones with the bacteria?
So again, this is more research that needs to be done, because one of the theories for why Ocedax were found on the non-lipid rich bones is that maybe the lipid actually inhibits them breaking down the collagen and getting the collagen. So maybe it's just easier for them to live on lipid poor bones. So there was an experiment devised where we boiled some whale bones to get all the oil out of it and then put those down, and then also put down some normal ones to see whether Ocedax would prefer one to the other. It hasn't been done yet, but it is because the Ocedax aren't relying on the lipids themselves. It does make sense that they would still colonize the bones, whereas the bacterial mats do need those sulfides, which are as a result of the lipids, and so they will only be found on bones where there are lipids and sulfides. So, does that answer your question?
So,
What was the question?
do you think when you go back this year,
Right.
that
Because the lipids have decreased, there'll be less Ocedax.
less bacteria, but the Ocedax will move on to the
Yeah,
cordial
maybe,
bones?
exactly. That is a very good hypothesis, that because they're now less lipids, yes, bacterial mats have decreased, and now Ocedax have spread throughout the skeleton. We don't know. We'll see. Sorry. Any other questions?
I might just curb the questions there.
Sorry.
But if you have questions, I'm
Come
sure
and speak to me.
Diva will be happy to take them afterwards. Just before we thank Diva for her presentation tonight, I just want to say thank you very much for your patience with being in this room this evening. I know the seating wasn't ideal, and I hope that next month we'll be back in the lecture theatre next door. And I want to invite you all to come to November's talk, which will be Clive Truman speaking about the life histories of fish in our waters, both continental shelf and deep, and that's on the 1st of November. So watch your email for that. So thanks, Diva.
And thank you for coming.